Vascular tissue consists of xylem and phloem, which runs through the roots, stems, and leaves of vascular plants. The function of xylem is the conduction of water and minerals from the soil to the leaves of plants. In gymnosperms, xylem consists of cells called tracheids. In angiosperms, xylem consists of two types of cells, tracheids and vessel elements. Tracheids and vessel elements begin as living cells, but as they mature, their living contents die, leaving the non-living cell walls in place. Vessel elements have thick lignified secondary walls. Tracheids taper at the ends and connect to one another to form a continuous column. Likewise, the vessel elements are connected end to end to form a continuous column. The ends of the vessel elements have perforations. Both tracheids and vessel elements have pores known as pits. Water moves through tracheids only through pits, whereas water flows from one vessel element to another vessel element through perforations and through pits. The function of phloem is the movement of organic molecules from the leaves to the stem and root of the plant. It consists of two types of cells, sieve tube elements and companion cells. Both of the cells are alive at maturity. Sieve tube elements have no nuclei. They have plates at both ends that are perforated with holes. Each sieve tube element has an associated companion cell that has a nucleus and carry out functions to maintain both cells. Meristematic tissue. There are three types of meristems: apical meristems, lateral meristems, and intercalary meristems. This image shows two types of meristems: apical meristems and lateral meristems. Apical meristems are located at or near the tips of roots and shoots. As new cells form in the meristems, the roots and shoots will increase in length. This vertical growth is also known as primary growth. As the apical meristem grows and forms leaves, it leaves behind a region of meristematic cells at the node between the stem and the leaf. These axillary buds are dormant, inhibited by auxin produced by the apical meristem, which is known as apical dominance. If the apical meristem was removed or has grown a sufficient distance away from an axillary bud, the axillary bud may become activated, or more appropriately, freed from hormone inhibition. Like the apical meristem, axillary buds can develop into a stem or flower. Each apical meristem will produce embryo leaves and buds, as well as three types of primary meristems: protoderm, procambium, and ground meristem. Protoderm develops into the epidermis. Procambium develops into primary phloem and primary xylem, and produces the lateral meristems. In roots, the procambium also gives rise to the pericycle, which produces lateral roots in dicots. Ground meristem develops into the cortex and pith. Lateral meristems. Lateral meristems account for secondary growth in plants. Secondary growth is generally horizontal growth or sideways growth. There are two types of lateral meristems: the vascular cambium and cork cambium. The vascular cambium is a thin branching cylinder that, except for the tips where the apical meristem are located, runs the length of the roots and stems of most perennial plants and many herbaceous annuals. It is responsible for the production of cells and tissues that increase the thickness or girth of the plant. In other words, it produces the secondary phloem and the secondary xylem. The cork cambium is also a thin cylinder that runs the length of roots and stem. However, it is found only in woody plants, as it produces the outer bark. Both the vascular cambium and the cork cambium, if present, will begin to produce cells and tissues only after the primary tissues produced by the apical meristem have begun to mature. Intercalary meristem. Intercalary meristems are found along the stem and at the base of leaf blades in grass and horsetails that do not have a vascular cambium or a cork cambium, as they do not increase in girth. Intercalary meristem at the base of most grass leaf blades allow damaged leaves to rapidly regrow.